Hello everyone, in this video we are going to talk about Sanger technique that he made a very nice and very excellent technique in order to do the DNA sequencing. So let's watch the video and see how does this work.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain.
Okay, I'm going to stop here because it is very important to understand what is going on during this process. This is a DNA sequencing technique, which is one of the most important facilities if you go to a lab having this DNA sequencer machine. So you need to know what is the technique and how does it work. This is the template or the DNA that you are going to have it sequenced, which means that as a question mark, meaning that you don't know what is the sequence, what are the order of the nucleotides, so that you don't know. How do you want to find it? It's based on the technique which is Deoxyribonucleic acid fluorescence, which means that those deoxyribonucleic acids has each of them has one particular nucleotide. For example, in this case it is G, and whenever the G is appearing in your sequence, they stop the replication process. Is it clear? So let's say the replication process and the copying from the original template or original DNA is starting, and then whenever it reaches to G, then the replication will be stopped. And the number of the G's, it doesn't matter how many G's is it present in this case, but the replication is stopping for any time that this G, which is a very specific G, we are adding to our we are adding to our machine, and we are adding to our YouTube, not YouTube, we are adding to our tubes, sorry. So funny. So we are adding to our tubes. We have these G's, once they are reaching to their positions in the replication, they stop their replication process. But we have separate and different fragments each time that the G will be added for replication.
This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the...
Okay, did you see that? This is so beautiful. We provide 4 different types of the nucleotides as deoxynucleotides or deoxyribonucleic acids, and they are specific and special, and also they are bound to a fluorescent dye which can be recognized or which can be identified through the machine later on. But the thing is, all these different nucleotides or all these deoxyribonucleotides, they are ending the fragment whenever they are attaching. Whenever they are coming, they end the fragment. So we have the different fragment sizes which which machine can recognize, and they all put together and makes it with this analyzing where is the C, where is the A, and where is the T, and the machine automatically put them a software which is inside the machine, and there is a computer connected to the machine, so everything goes together, and just like boom, that's a magic, so you get your DNA sequence. Let's continue.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
Okay, we go back to our slide. Here we go. Deoxyribonucleic acid stops the DNA replication when it is added to a new DNA strand. So these are the 4 deoxyribonucleic acid here. Okay. And you see that how it has been written D-deoxyribonucleic acid. So we have DD or D-deoxy-ATP, D-deoxy-GTP, D-deoxy-CTP, and D-deoxy-TTP. Each of them represent one of the bases: adenine, guanine, cytosine, and thymine. So, and together with this, we also have our tubes, including DNA polymerase. Also, we have the regular DATP, GTP, and CTP and TTP. So these are the regular one because we need to have their each time we need to add only one of these in order to make different fragments, and limiting amounts of fluorescence label, which is these deoxyribonucleotides. So once the replication is starting, when it reaches to here, which is T, then the fragment stops, the replication stops. Again, another starting replication, again it reaches to T, then it stops, and it reaches to G, then it stops. It reaches to T here, and again it stops. And for each of them, we have a different size of a fragment regularly, that it will increase the size from here to here because it goes to the complete length of the DNA that is going to be replicated completely. So we have a complete different size, and the next step is, in this technique, these different fragments in the machine are going to be loaded in an electrophoresis. So you also run an electrophoresis after when you are running the machine, and in this electrophoresis, then you run it into the well. Well means the place that you add these the tubes that you have already got it from the mixing after PCR. So once you have run, then based on the different fragments, they separately sit according to the gel. So can you tell me which fragment shows the longer one, and which fragment shows the shorter one. Pause the video and answer this question. This is very important for your understanding. Okay, I suppose you paused the video. Yeah, I think you did. So, what was the answer? Yes. You know that in electrophorescence, the fragment which is heavier, it will be in the top, and if the running gel showing like this, then the one which is lighter is going farther because it is light and go farther in the electrophorescence, and the one which is heavier, it will be kept at the top. So as you can see, the sequence will be shown up like here. This fragment is T, is lighter, then it will be here, then the next one which is lighter, then it is here, and the next one TTG, and the next one is G, TT, then G, and the next one again you can see is T, so it's T. So based on the size of the fragment, then it will go step by step down, and the one that is lighter goes in the bottom, and the one which is the heaviest one, which is G, as you can see, this is G, and the fragment is the heaviest one, so it will be in the top. Let's read these together. Fluorescent dye markers are attached to deoxyribonucleic acid so that the base present when replication stops can be identified. From this, the base and the parent strand deduced. DNA replication is carried out with deoxyribonucleic acid. Mix it with normal deoxyribonucleic acid. Yeah, so this is what you got in. It's not that everything here with the fluorescent markers, because we need a little one markers as fluorescently to make different fragments, different sizes once they are added. The length of a strand and the terminal base are identified by sequencing machine, as I showed you how. Okay, let's continue with the second beautiful topic on the molecular biology, which is DNA profiling. If you remember, we mentioned that we have coding and non-coding regions in the DNA. And what is the purpose of non-coding regions? Can you pause the video and think about it a little? Yep. So I think you paused the video. So there are non-coding regions. These non-coding regions means they are not translated to proteins, but they can be used for other purposes. One of the other purposes for these non-coding regions is that they have a tandem repeat sequences, or the sequences that they are repeatedly can be observed as a repeats. For example, can you see that? What are the repeats over here? Gata, 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 gata. So how many gata? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yes, so we have 10 gata here, but we have 9 gata here. We have 8 gata here. Why? This is the reason that these non-coding areas can be different between people, between man to man, between human beings, and this is the key for finding the differences between two people. And that's why we can check our DNA and see, oh, this is not you, or this is not your son, this is not your daughter, this is not the person you killed, and because your sample taken from your blood is not matching with the sample was taken from the crime scene. Oh, thanks God. So that's it. How these tandem repeats are used in DNA profiling. Okay, this is the video. I have already downloaded this video on ManageBack. You can go and watch this. If you have problem with downloading from ManageBack, then I will put it in Baidu app as Baidu link as well. Don't worry about that. So this is a 13 minutes video. You have to watch it by yourself. I'm not going to repeat it again here, but I'm explaining that these tandem repeats greatly in terms of different number of copies of the repeat element that can occur in the population. So we have the different repeats in the population, so that's why we can separate and we can differentiate between two people and if they are matching or not. One of those very important experiments which is used by these tandem repeats for maternal profiling. It is usually mitochondrial DNA. So they take mitochondrial DNA for maternal profiling. For paternal profiling, they use the Y chromosome for showing that if it's your dad or not. And if it's your mom, then mitochondrial DNA. If it's your dad, your Y chromosome. So these tandem repeats are mostly the same between you and your father, but it is not the same between like your father and with the other person, another guy. So these tandem repeats are not the same between two people, but mostly are the same between two very close genetically people, like father and son, mother and daughter, or mother and son. That's it. And dye markers attached to the deoxyribonucleic acids are attached to the tandem repeats during the PCR. This is the techniques, how it's done. It's not the concept, it's the technique. It's exactly the techniques is explaining what we talked in previous slide regarding how the DNA sequencing is done. So this is definitely is based on the DNA sequencing. This also not only can be done using DNA sequencing over here by going back to your previous slide, you will see how, but also it can be done by the restriction enzymes. This restriction enzyme can cut the DNA and see if the tandem repeats are the same between two people. So here is the DNA and the restriction enzyme. The restriction enzyme can cut the DNA at these areas, and if two people have exactly the same number of the tandem repeats, then the restriction enzyme can create exactly the same fragments with the same size. But if you have the different numbers of the tandem repeats, I don't know what this line is coming out. It's really weird. So if you have different tandem repeats, then these sizes are not the same. So by cutting the DNA with the specific places which are the restriction enzyme, you get exactly the same sizes of the DNA cut, or different sizes. That's another technique. So the first one is exactly the sequencing techniques. The second one, the restriction enzyme technique. The third one, electrophoresis, enables scientists therefore to calculate the length of the tandem repeat sequences. This is not the separate technique. It's the way that you, after cutting the DNA, then you should do the electrophoresis to see the size of the DNA. And if different tandem repeats at different loci are used, then a unique profile for an individual can be identified. So if you get exactly the same sizes of DNA cut or DNA fragments, then they are the same people. I mean, the son and father, for example. If you get different sizes, then that would be different people. And I think this is the last part regarding this DNA profiling. These tandem repeats also may have another function, like the end of chromosomes which have highly repetitive DNA sequences. These ends are called telomeres. These are telomeres. These are responsible for not allowing the degradation of the molecule during the replication. So this is also another function for these tandem repeats. And because they are not copied and they are not translated, so it is not important if they are getting damaged or if they are, something happened to them, because they are not responsible for making translation for proteins. But with their number of the repeats, they can protect the chromosome, and they have a protective role for not degradation of the DNA molecule during the replication. And that's it. And we reach to the final slide of the 7.1. And congratulations!